All right, hello students. This is a fresh uh, lecture video for Chapter 18 in the OpenStax Biology 2nd Edition textbook, which I've got the cover page for right here. And we're going to go over Chapter 18, which is the Evolution and Origin of Species, material that we've already discussed a little bit and quizzed on some. So, a few things right here. You do need to know about Darwin, and if you go to the study guide, which I'll take a look at the end, there's some additional people that we need to talk about. And we always start with a review of Charles Darwin's work in here because he's most closely associated in popular culture with evolutionary biology, and he is important scientifically in that because he's the first person who put together a really clear and coherent, and as far as continued experimentation shows, largely correct explanation for how species could change over time, and by extension, how we've gotten to the biological diversity that we have on the planet right now, which is to say biological diversity is all the different species of all the different stuff that we've got out there: bacteria, lower plants, higher plants, animals, et cetera, all that diversity, as well as the diversity within any one species or within any one population of one species. That's all Darwinian evolution due to natural selection. Darwin, of course, did his work on a Royal Navy ship called the Beagle in the early 1800s, 1830s, is when he did, late 20s, early 30s is when he did his most salient, research as a relatively young man. And then he spent quite a few years, even decades, working and chewing and mulling over the terrific amount of information that he collected, the huge amount of data, the terrific number of specimens and all of that he collected in his voyage, before finally writing a most famous book on the origin of species. So the main ideas right here are, what he observed and, may hopefully seem fairly straightforward to us today. Most characteristics are heritable. So in other words, the phenotype, the thing that you look at when you look at an organism, or oftentimes far more important, the biochemical characteristics, what versions of digestive proteins they produced, what kind of immune proteins they produce, how well they metabolize this particular sugar, whatever it may be, all the various proteins that are the result of expression of genes, all of that is heritable material. And there's quite a bit, and so there's a huge amount of that material, that defines what an organism is, that can be passed from parent to offspring. It is preserved in the germline through the DNA code. He didn't know anything about DNA, but everybody understood that somehow some sort of information went from one generation to another. Another huge idea of his is that more offspring are produced than can survive. And so this sets up a situation where there's going to be competition for those resources. Not everybody is going to have access to all the resources they need to survive, and some will not survive and some will. So the hard cold truth that we're largely spared of, certainly here in Fort Worth, in Texas, in America, and in the modern age as humans, is that most offspring die of every living species. Some have a better chance of living till old age. A baby elephant, for instance, whereas, a baby trout, has an incredibly low chance of, making it to adulthood. And that's why trout have thousands, if not hundreds of thousands of eggs that they lay, because, so few of them will make it to adulthood. There's so many different ways in which that works out. But the long and short of it is, inevitably, populations are going to reach a point as they grow where more offspring are produced than can survive. And so this sets up a competition, which is the key idea of natural selection. Natural selection is really a winnowing process where the environment punishes those who cannot get at the resources they need to live and rewards, those who can, with the ability to reproduce. There's nothing sentient going on here. It's just the nature of the way it goes with things that reproduce exponentially and that have variation. any system that does that is going to lead to a selection process. All right, and that includes very definitely biological systems. All right, those characteristics in the offspring vary quite a bit, and those variations are heritable, right? So, characteristics are heritable, passed on from one generation to another. Many more offspring produced than can survive, so this sets up the situation of competition for resources, and that characteristics of offspring vary, even from asexually reproducing things like bacteria. They normally clone themselves, they make exact copies of each other, but when that random mutation shows up, we've got variation, and that variation typically is heritable. The result is this leads to the phenomenon of adaptation, where varying characteristics that make it more likely that organisms can get at the resources they need to, is going to be rewarded with increased survival and increased reproduction. And that is the accumulation of those changes is what we mean by heritable adaptation, as opposed to the adaptation you get by running every day and getting better at running and having, more cardiovascular capacity. That is, reversible adaptation just within your own body. That's not heritable. That's a different thing. All right. So Darwin, famously is associated with the finches of the Galapagos Islands. There's so much other work he did, but, for the sake of brevity, we just pick a few here, to point at. But he did some of his most rigorous work with these particular animals in this situation. So the idea with the finches is that one species of finch, maybe something looking a bit like this or perhaps like this right here, but not any of these, none of these 4 species, is the parent species of finches, and it lives in Ecuador. It lives, in South America, and for whatever reasons, a flock of those finches, or at least 2 individuals, a male and a female, or, even possibly, although there's a fair amount of genetic variation in these populations, so it kind of suggests a large number made it, but even possibly one female who was already pregnant, if you will, had fertilized eggs in her, and when she arrived at the Galapagos Islands, had a clutch of young that survived, and, at least one male and female were in there. Theoretically, that could start a population. If there were not any terrible recessive genes that caused major defects present in her, then her offspring, might be good enough to actually start a population of all birds that would be, identical to each other. More likely a flock arrived on one or more of these islands. And these are birds that don't normally fly far across the ocean. And there's plenty of evidence of this. This has happened again and again in modern times. It's been demonstrated. But clearly what would have happened is a flock of these guys would have been driven offshore in a storm. They're not going to land in the ocean because they're terrestrial birds. They would just die. And so they keep flying and, eventually died and fell into the ocean or got exhausted and landed and, drowned instantly. But every once in a while, one of those flocks, finds itself over an island, and you bet they're going to go straight down to that island and sit down and never fly again if they can help it, if it's possible. Which is why we've got species of flightless birds all over islands all the way around the world that are related to flying birds back on some larger mainland somewhere. The reason they don't fly anymore is because, if they arrive and there's no predators and there's no trees, nesting is going to have to be on the ground if they're going to survive. And, living on the ground without negative consequences tends to select for bigger birds that are stronger, that can get more food, maybe dig holes for their nests, whatever it may be. And flight, is usually a bad thing because they're on an island. They don't need to go far. Flying gets you blown offshore into the ocean again. And, et cetera, et cetera. It doesn't mean it will happen, but that's an explanation for why that's so current. Anyway, back to the story here. So a population, or at least one, pregnant finch arrived on one of these islands sometime back in history, and for the same reasons that they ended up from the mainland on one of these islands, over time they diversified to these other islands, getting blown over there, landing, never flying back to where they came from. And they eventually populated all the islands of the Galapagos. It is a archipelago, a series of islands just, that are visible to each other, but far enough apart that, small birds don't like flying from one to another. And so we have all these different species, and the species are adapted to their local conditions, primarily the foods that are available. So this is a bird that eats big, tough fruit and seeds. These are seed-eating birds primarily. They'll eat some insects and they'll eat, fruit and things like that too, but seeds are their sort of core food. And these guys are going to be adapted for eating really, really tough, seeds, while these guys are going to be adapted for picking tiny little, very delicate seeds, or even going after insects and things like that, and various other specialties. Birds, the food they eat is telegraphed in the beaks of birds, because they're highly varied based on what they are adapted to, eating in their environment. All right. A couple of names you want to know. So Darwin and Alfred Wallace are both people who really put together a terrific body of observations, created the hypotheses, tested them with observational, data collection, and basically wrote the same theory, or the same ideas down. There were hypotheses that required testing, and their data collection was part of their testing of that, that are natural selection. So they really came up with the same idea together, and they presented them roughly at the same time to the Linnean Society, which is the one of the components of the Royal Society in England, the equivalent of the National Academy of Sciences here in America today. They did this back in the 1850s. It's famously, Wallace knew Darwin and knew Darwin's work. He was working over in Indonesia, spent a long time over there, and he sent his manuscript on, the change in populations over time due to natural selection to Darwin to see what he thought of it. And Darwin went, "This is exactly what I've been working on all this time." And so he was finally spurred by Wallace's sending him his manuscript to finish his book on the origin of species. And then, thank heavens, he was in England. He was over in Indonesia, so not there to defend himself. Darwin did not steal his work, but instead presented for, on both of their behalves, if I remember correctly. But anyway, was very magnanimous to, to share credit, for this work with Wallace. But Darwin kind of goes down in history as the person that we most associate it with this work. So, evidence for evolution. There's, some things that we are going to take a look at. These are, 6 different sort of major sources of evolution that are the sort of stuff, at least a lot of it was the sort of stuff that Darwin and Wallace would have worked at. Some of it is, dependent on more modern scientific techniques, particularly having to do with molecular biology and early developmental features, as we go from egg to multicellular to an embryo and all of that stuff. All right, so we'll take a look at each one of these. All right. So first, let's compare, let's look at this word selection. There are 3 different types of selection that occur out there: natural selection, artificial selection, and sexual selection. All right. And all of them are types of selection where some individuals have a better chance of reproducing and others do not. Natural selection, based on their, their innate abilities to get at food, to survive long enough to reproduce. Artificial selection, people decide who reproduces. And in sexual reproduction, the potential mate or competitors for a mate, depending on the particular form of sexual selection, determine who gets to reproduce and who does not. And it's one of the reasons male deer have great big antlers. That is sexual selection at work, where males compete with each other and females only choose, winners of competitions. All right, so that's sexual selection. There are 3 types. All right. Homologous structures. This is one of the things that, the more you study biology, the more detailed information we get on these ideas right here. So, homologous structures are structures that exist in 2 different species or groups of organisms that are adapted, modified for new tasks or for different tasks, but clearly have, a homo, meaning same, origin pattern. And the implication here is that they really do come from a common ancestor, that these are modifications of something some ancestor of both of these groups had, and that has been modified, for the evolutionary trajectory, the environmental selective pressure, and other selective pressures that these organisms faced. So here's the forelimb of 4 different vertebrates right here, and they're all, well, they're not all mammals, but 4 different vertebrates. And these are part of the argument that all vertebrates are descended from a common ancestor, because if you look at their forelimb, the front limb or the upper limb in the case of something that walks upright, we see that they have this pattern, and the rear limbs have a very similar pattern. Big bone, 2 bones, pile of little bones, and then a set of rays, most typically 5 or some reduced number from 5 of these multi-bone, final pieces right here. But 1 bone, 2 bones, a pile of little bones, and then these rays show up over and over and over again. And, this of course is the humerus, this is the radius, this is the ulna, these are the carpals, and these are the metacarpals and phalanges of the hand. This is the foreleg of a dog, and again we've got the carpals, the metacarpals, and the phalanges right here. The humerus, the radius, and the ulna, etc., etc. On a bird, the 5 rays have been reduced substantially. Here on a whale, they're still present, but they're buried in a great big gigantic flipper. But all the bones are there, and so homologous structures, we see that all over the place, and there are so many homologies, but this is one of the more dramatic ones. And so the idea here is that anytime you see this pattern in a bunch of organisms, there is a strong suggestion that they are modifications of a shared common structure, which would have been the front walking limb of a terrestrial vertebrate that is the ancestor of birds, that is the ancestor of whales, and that is the ancestor of humans, and of course the modern dog. And the dog retains the most original version of this limb, and the others have highly modified versions of this limb for the worlds they live in now and are adapted to. Okay, on the other hand, and that is a case of divergence. So this common structure has been divergent evolution, to solve local problems. Carrying things and brachiating. Our arm is the result of our ancestors brachiating, moving through trees, and their forelimbs are the primary, means of moving around in an arboreal setting, while the rear limbs, are used for moving around on the ground, as we see in most primates today. But a lot of the primates are knuckle walkers. They'll use their fore and their rear limbs while walking on the ground. But chimpanzees can stand up badly on their rear legs, just like gorillas can, etc., etc. All right, and of course modified for flight, modified for swimming. Convergent evolution, let me adjust this a little bit right here. Sorry, hang on a second. There we go. Convergent evolution is one of the confounding processes right here. And so, if you look at convergent features, you might categorize things together because they share characteristics that are just results of very different organisms from very, very different reproductive origins, from ancestors coming from very different places, converging, which means to come together on the same solution to a shared problem. So for instance, in here we're looking at coloration. Because this dove is white and this fox is white and polar bears are white and a number of other things, Arctic terns are white, any number of animals, that means that this adaptation, this white coloration right here, is typically actually an adaptation to make them harder to spot, against a white background of snow and ice. In this case, to avoid predation by bigger predators and to hide from prey. In this case, pretty much strictly to hide from prey. But the fact that they're white is a shared solution to the same problem. How do you avoid standing out against a white background? There's really only one solution, well, 2. Invisible, which there's not much way of living things doing that, or being the same color as snow and ice, and that's white. So the fact that they are both white does not imply that they are particularly closely related to each other, and we'll agree this is a mammal, this is a bird, yet they both share this superficial feature. Whales and fish have a number of convergent features that make them look really similar to each other, and and even function similarly to each other, and those are adaptations that make it possible to swim through the water quickly. All right, the shape of the tail, the shape of the body, the surface texture, and any number of other features. But of course, fish with gills and whales with mammals really have two completely different evolutionary origins. In fact, whales are descended very, very demonstrably from terrestrial animals. Their ancestors lived on land. And, of course, currently their whales live in water and superficially look like fish. A long time ago, people called them fish. Biogeography. This is the idea that the surface of the earth, the geography, where there is land, where there is water, where there are mountains, where it is cold, where it is hot, where it is wet, where it is dry, all of those things have a huge impact on how life is distributed around the planet today. So, for instance, earlier I was talking about Darwin's finches, and the biogeographical information that's required there is that South America has been around for a good long while. All right. It was part of Pangaea originally. South America ended up being part of the southern continent that broke away from Pangaea, called Gondwana land. That was South America, Africa, Antarctica, and India, and Australia. That has broken up in turn, and those continents have gone all in their various directions. Well, anyway, South America has been around for a while, though. It was floating along separately, not connected to North America until relatively recently. Finches lived all over South America and have for quite a long time. They evolved, over the last 20 million years or so, and are a diverse group of birds, along with all the other birds, that are in South America. The Galapagos Islands are much, much more recent. They only appeared, hang on a second right here. They only appeared about 4 million years ago. So much, much more recently than South America, which has existed, as a landmass and separately, all on its own, disconnected from other things for well over 100 million years, 100 million years. So the Galapagos are fairly recent. So sometime, after 4 million years ago, when the Galapagos Islands erupted from the ocean as volcanoes, just like Hawaii is still doing today, same type of origin, those finches, flew for whatever reasons, probably blown by a storm is the most likely answer, from South America out to the Galapagos Islands. All right, that is a biogeographical event, and then distributed across those islands and diversified into the various species that we see today over the last 4 million years. That's a case of biogeography. Similarly, when Pangaea broke up, the southern continent was populated by marsupial mammals. So there were reptiles and all kinds of other exciting things back then. And this is still back during the age of dinosaurs. But some of the early mammals that were on Pangaea, very early on, very primitive mammals, were divided up into two big groups, the northern continent and the southern continent. And the marsupials only, as far as we know, diversified and were really prolific on those southern continents, Gondwana land, after it broke and separated away from the northern continents. Placental mammals developed in the northern portion, which we are descended from. And these continents have moved around. The ones in Antarctica all went extinct because Antarctica, moved to the southern part of the planet over time. That is a biogeographic event itself. And everything went extinct, that lived on what's now Antarctica. It used to be a tropical, land. South America eventually made contact with North America, and most of the placental mammals in North America wiped out the marsupials in South America, with the exception of seven species of possum or opossum. And in Australia, no placental mammals have ever, until very recently, have arrived. It was all on its own for ever and ever and ever until very recently, historically, evolutionarily and geologically, and even more recently, due to human influence, did, only recently placental mammals start showing up in big numbers in Australia, and that'd be dogs, cats, goats, rats, mice, and all of those things, which are out competing the marsupials there and knocking a whole bunch of them towards extinction. All of that is stories of biogeography. The world is changing. The continents are moving. They take things with them. They bring things together. They separate them. And that has huge impacts on how species can evolve over time. Then, of course, much more recently, we are introduced to molecular biology techniques, and that is primarily looking at DNA. And so remember, from 1406, DNA, deoxyribonucleic acid, is the stored information on the chromosomes, of every living organism in the form of genes. Those genes code for RNA. A lot of that RNA, it turns out to be messenger RNA that codes for a protein out in transcription and in translation. And those proteins, are the phenotype, define the phenotype of the organism. And of course, mutations in those genes can lead to mutations in proteins that evolution and natural selection can act on. A thing about DNA is that it gives us lots and lots of this. So if we look at the same gene in 4 related organisms, we can oftentimes, see that there are, variations. There are letter variations in the DNA sequences that code for a protein in 4, different species right here. And, by doing some analysis of that material, we can identify, the what would have been the common gene that was the ancestor of all of these. And for instance, mutation-wise, what you're going to find is that, the genetic coding for this version of the limb right here, whatever proteins are responsible for developing this leg right here, those genes are going to be most like the ancestral genes of all 4 of these groups. You'll find that these guys, the genes that code for the developmental cues that lead to these limbs, and it's genes, coding for proteins that determine how frequently cells reproduce, how long they get, how long they last, and all of that stuff. Those genes are going to be a little bit more different than the dog's is, right? They're all going to be different in their own ways, with this being common. And if we look at other species that have that have legs like dogs, like terrestrial mammals walking on all 4 limbs, we'll find that the genetic basis for that is more similar amongst them than it is to these highly derived groups right here that have very, very different, have had some major changes in their structure due to genetic changes. Similarly, DNA can converge. 2 genes that are under selective pressure because the proteins that they code for have a big impact on, what's happening with the species out there in the real world. Mutations that make those genes create the same thing, oftentimes those mutations are going to look the same. Now, we can have different mutations lead to the same change in phenotypic expression. That can happen, and that would be a clear case of convergence. And oftentimes, so you can have the same mutations in a gene leading to the same phenotypic change, or there are other possibilities. We can change just this amino acid over here in the sequence, and it still alters the proteins, so that we get the same phenotypic change. Lack of pigment, pale hair, so there's lots of convergence in genetics that we can see and identify as we get better and better at working with genetic material, with molecular material. And we can work with DNA, we can work with RNA, we can work with proteins. All of that is available for analysis and consideration in assessing what's happening evolutionarily between organisms. Okay, some major ideas that I want to get taken care of right here. So let's talk about this really quickly. From 1406, hopefully you'll recall when you studied the scientific method all the way back at the beginning of the semester, you would have talked about hypotheses, theories, and laws. So, in common language, a theory, of course, just means I've got a theory. I've got a theory about that, and it's based on some information, but oftentimes what we're kind of talking about is a hunch, a weakly supported idea that we can test. We can see if this is the answer to the problem. In science, a hypothesis is a fairly well-supported theoretical explanation for something. Right? And we don't just pull it out of the air. We don't go, I'm going to guess this is it. It's based on a lot of stuff that we know and a lot of observations that we make. And that leads to a hypothesis, all right? Then we test that hypothesis. We run experiments or we go and collect information, not experimental, but observational science like Darwin did, instead of manipulating something. but we collect information to test that hypothesis. And if we test that hypothesis and keep finding support for the hypothesis, yes, the predictions of the hypothesis were met with this test, or this observation, or this experiment, we keep accumulating that. Eventually, we're going to go ahead and change the name of that hypothesis. We're going to go ahead and call it a theory, because it is a well-supported, experimentally supported. We never say prove, because you can always find something that might change our understanding of something, but extremely well-supported hypothesis. And that's what evolution is. The theory of evolution is an extraordinarily well-supported hypothesis that works to explain a huge amount of the biology that we see out there, particularly about how things are related to each other. All right. Next, so evolution, yes, it's a theory. It's not just a theory. That's an incredibly important thing. Now, laws are a totally different thing. In science, a law is a measurable thing where you can go out and observe it, and it always acts this way. The law of gravitation, Maxwell's equations of thermodynamics, the electromagnetic laws. There's only a short list of them, and those are like physical constants, that always work this way. This much mass will always generate this much gravitational attraction. Those things are the things that we define as laws. All right, and it's usually the law refers to the mathematical, the way that we can describe that thing happening. The inverse square law of distance for gravitation as an example. Okay, individuals evolve. We do not. Evolution depends on me attempting to make a copy of myself, however I do. Asexual reproduction, Biology 1406, or sexual reproduction, where two individuals are required, again, Biology 1406 material. But I'm attempting to make a copy of myself. And for evolution to work, the copies can't be identical to me. There's got to be variation based on mutation in those individuals, and then the environment selects those individuals. So individuals do not evolve. A population evolves as it reproduces over time. Evolution does not explain the origin of life, although there's certainly hypotheses that come out of evolutionary theory, that go back. But what evolution really explains is what's happened since life appeared on Earth and how it has diversified from the early fossil record of bacteria all the way through to the, startling diversity of life that we have on Earth today. And I've talked about this before, I think, in our class section. There is no evolution on purpose. Aside from ourselves, there's no evidence that any organism knows, in a way that could influence, which individuals get to mate and produce offspring to, prepare themselves for the future that's coming. All right. It doesn't mean that there's not other species on Earth that are aware of themselves and aware of the future. In the past, there's a lot of evidence that many of our colleague vertebrates, for sure, and perhaps certain invertebrates also, have that level of complex thinking. But we're the only ones who are aware of this idea to our knowledge. So we could make ourselves evolve purposely by governing what happens with our gene pool over time. It's a huge source of interesting philosophical and moral ethical dilemmas, big stuff outside of science, really. Or, that science can't solve. Doesn't mean it's not important to science, but it's stuff that we can't solve, ourselves. Is it good or bad, to, change our species. Anyway, so back to the point is here, mutations occur when they happen, and if those mutations are, significant in a selective world, in natural selection, conditions, then the species can change over time, and that's it. So, the accumulation of resistance to diseases, that lots of bacteria, accumulation of resistance to antibiotics, the accumulation of resistance to poisons by mosquitoes, and things like that, those have all happened because you had big populations of organisms with a lot of variability, and that variability was always present, or was present before the selective event, antibiotics or poisons against mosquitoes, started showing up in the environment. And so we killed off a lot of individuals, but those that possessed the variation, necessary to allow them to stay alive under this extremely selective environment of antibiotics or poisons are the ones that are left today. So now we have poison-resistant mosquitoes and other pests and antibiotic-resistant bacteria. If the mutations weren't there in time, theoretically every individual could have gone extinct, could have been killed off, and we could have killed off a species of bacteria or killed off a species of, pest like mosquitoes. Formation of new species. So, there are some interesting questions right here. The key definition: a species is a unique gene pool that does not have genetic material routinely coming from another population. So it's all the individuals that share a gene pool that's distinct and different from other gene pools out there. What do I mean? All of humans have a gene pool, right? We have genes that code for all kinds of features where we have different alleles. They're out there: darker pigmentation, lighter pigmentation, taller, shorter, heavy bodies, skinny bodies, whatever it may be. Lots of hair, not much hair, curly hair, straight hair, all that kind of stuff. That's all part of our human gene pool, all right? There's a gene pool for all of the other great apes: chimpanzees, bonobos, which are a separate species of chimpanzee, the smaller, more social ones, gibbons, this three or four extant species of gorilla, lowland, mountain, eastern mountain, etc., etc. They all have different gene pools, and there's no sharing of genetics, or at least not much, between those gene pools, because there's no reproduction between these groups. I say not much sharing of genetic material because there's vectors like diseases, viruses, and things like that that can move genetic material from one species to another. They're rare. They're incredibly important in evolutionary biology, but not important in this conversation. Okay, and that's what we mean by a species. So a species is really an isolated, separate gene pool. It is most useful as a definition when we're talking about sexually reproducing organisms, where it takes two individuals, sharing a combination of gametes to produce a new individual. That's the most robust, version of the species definition we can have, and that's what we call a biological species. So asexually reproducing things: bacteria, asexually reproducing animals, asexually reproducing plants. We call them species, but basically every lineage is kind of its own species in a sense, since there's never any interbreeding amongst individuals. And then we've got a huge pile of things that are both sexual and asexual out there. So they reproduce both ways in different conditions. All of them would be true biological species in our conversation. So things that can hybridize, when we have hybridization between two things that we call species, if we get a viable hybrid that can reproduce itself, that really says we don't have two species, we have one species. If we get a hybrid that can't reproduce, then that is the product of two distinct species. So a mule is a hybrid. It's the product of cross of hybridization between a horse and a donkey. You get a mule, but the mule itself is sterile. So that means horses and donkeys are separate species. They're just highly compatible. You can get a hybrid of a wolf and a dog, and it is a wolf-dog hybrid, and it can reproduce, with wolves, it can reproduce with dogs, and that means that wolves and dogs are really the same species, and they are. They're just two very, very isolated gene pools. In the wild, normally wolves kill dogs, occasionally the other way around, but they also can and do mate under certain conditions. So, it makes the gene pools not fully separated from each other. All right. Two big terms right here. Speciation. So we're moving from what happens within a species to what happens amongst populations right here. So this is, going into chapter 19 material. We'll come back to it, and there's more of this in the next chapter. But this idea of speciation. So if you separate two populations one way or another, so their gene pools are isolated from each other, you have the potential for speciation. All right, and two gene pools that don't share any genetic material really are, by definition, two different species. If hybridization is completely impossible for whatever reasons, then we've absolutely got two separate species. All right. Okay. There are two ways in which this generally happens. There's two patterns that we see out there that explain pretty much all examples we might encounter. One is allopatric speciation. That means in a separate place. And sympatric speciation, which means in the same place. So what this means is the two populations are separated, usually geographically. All right, they're separated either because a population went off by itself, Darwin's finches, from Ecuador landing in the Galapagos Islands. That's a dispersal event, and they never went back to Ecuador. The ones that arrived on the Galapagos Islands, to our knowledge, and so they were isolated. And since then, they have diverged, substantially into unique species that are not going to hybridize with their colleagues back in Ecuador. Vicariants, where the world splits in half, the ancestors of monotremes and marsupial mammals in the southern hemisphere of Pangaea when it broke apart. That's a vicariance event. Everything, sailing off in the southern oceans all the way over to where Australia is today, that was a vicariant event. And, the animals that were on Australia evolved into their own unique, species, irrespective of what happened back in South America, what's now South America, and what happened in the northern hemisphere. And then finally, adaptive radiation, also Darwin's finches. Finches arrived on one island, found an environment with lots and lots of different ways to make a living. Some islands with lots of seed food, lots of insects, others with a really harsh environment, not many plants and not much stuff to eat, big seeds, little seeds, whatever, different climates, cold islands, wetter islands, drier islands. And the finches have separated into smaller populations on each one of those islands that have specialized for those particular islands' environments. And that is adaptive radiation. When new opportunities for diversification appear, and a species is able to take advantage of that opportunity and disperse, into these new areas, that event is called an adaptive radiation. And so, for instance, over history, over time on Earth, lots and lots of species have gone extinct, and every time they go extinct, the way they made a living and where they made a living and when they made a living, daytime, nighttime, summer, winter, all year long, whatever, becomes available for anything else that can take advantage of those opportunities. And the species that do that have adaptively radiated into that new opportunity. Birds and mammals took over all the roles that dinosaurs once played in the ecology that they lived in. That is an adaptive radiation event. All right. Sympatric speciation is where you get a new species right where the previous species that it came from existed. And this usually has to do with major changes in the way reproduction works, usually having to do with chromosomal changes. For sexual reproduction to work, the chromosome count has to be the same, 23 chromosomes for both individuals. Horses and donkeys have a different number of chromosomes, which is why they can't hybridize. Or when they do, the hybrid is sterile, I should say. The reason they can hybridize is because most of the chromosomes are the same, but there's a difference in numbers. All right, so aneuploidy, autopolyploidy, and allopolyploidy all refer to changes, as it says right here, in the chromosomal complement inside a cell. And typically they are the results of errors in some sort of process of cell division or DNA duplication. Aneuploidy is a change in the chromosome number. So for instance, one of the things that distinguishes humans from, let me see if I can get this up right here so we can see it, humans from chimpanzees is the difference in the number of chromosomes that we have right here. Here's our 23 distinct chromosomes. This is through 22 right here, and then here's the 23rd pair X and Y chromosomes that we're familiar with, all right? And then here's the mitochondrial genome, mitochondrial DNA right here. Only goes matrilineally from mother to all offspring, only from females to the next offspring, etc. That's what the M means here. The chimpanzee, they've got they are different in that their chromosome number 2, the chromosome number 2 that we shared with, that our ancestors shared with their ancestors way back, has split in two. And so they have 24 chromosomes, and then their X and Y pair right here, and then their mitochondrial. So that makes us completely different species. There's no way we can hybridize because you can't have sexual reproduction with these radically, this radical change right here in chromosome count. And it's the result of a break. It's clear that the material that was on chromosome number 2 is now divided up between these two other chromosomes. And, anyway, that's what we understand now from investigation. So that's a case of aneuploidy. So once two populations are isolated for whatever reasons, that process can occur, and that can happen in allopatric speciation as well. Oftentimes it does, and that's what prevents rehybridization later on when they get back together. When whatever reasons took them away from each other brings them back in contact with each other. Autopolyploidy is the case where an organism duplicates its entire genome set in one go. And that is what has happened with any number of plants. This is extraordinarily well known from plants. There are lots of plants that have not just 2 versions of each chromosome, but 4 or 6 or 8. And anytime it's an even number like that, odds are they've duplicated their entire genome. So sometimes that happens because in meiosis, we don't have separation of the homologs in meiosis 1. So a diploid cell stays a diploid cell. And if that happens in a single plant that still somehow produces eggs and sperm in its flower parts, or however, whatever type of plant it is, and those diploid cells fuse together, a diploid sperm and a diploid egg, we now have a quadruploid, not a diploid, but a quadruploid plant. And this has been seen. This has been shown to have occurred. It has been induced, and there's tons of evidence out there in the plant world for that happening. A variation on the same idea is that the second copy comes from an outside source. It doesn't, the extra copy of the chromosomes come from another individual. So that would be 2 different plants. This is much more typical in plants. Plants are much more tolerant of chromosomal weirdnesses in counts and numbers than animals are because of how they develop. So this is really, really common in plants. But allopolyploidy is the same sort of idea, except one individual has had this happen. They've duplicated the genome in one of their gametes, and that gamete still fused with, let's say, a haploid gamete from another plant, male plant over here, female plant over there, and they combine themselves together to create a triploid cell. And if if that happens twice in 2 different groups of plants, or in 2 different plants in 2 different locations, and then the gametes from those plants ever find each other, triploid gametes, we've now got a new species up and running that may have happened in the same field of, tens of thousands of flowers, flowering plants, of some plant right in the same field, and that's sympatric speciation. Interestingly, it does happen in animals sometimes too. The paper that you have to read on the crayfish, the new species of crayfish, is an example of really a sympatric speciation event. And it has to do with a change in the ploidy of the cells, of the reproductive cells, and a whole bunch of interesting things. Now finally, for both cases, allopatric or sympatric speciation, gene pools can be isolated from each other, based on things like what are called prezygotic barriers. That means before egg and sperm can come together. All right? So, say, individuals of one gene pool are only reproductively mature. Think of insects that just live a year in the fall, but there's another population where they're reproductively mature in the spring. All right, that's temporal isolation. Their gene pools will never meet each other. And then over time, chromosomal changes can occur that will lock in that difference between the 2 of them, because errors happen, and occasionally they take. That's an example of a prezygotic isolation, though. Habitat isolation, where the individuals living up high on the mountains don't really ever meet the individuals living down in the valleys of the same species. So we've got 2 separate populations reproductively isolated from each other. Largemouth bass, in 2 different lakes in Texas are habitat isolated from each other. Birds that only live in forest and won't fly across open fields are isolated in the little pockets of forest forests they have all over the place, which is one of the reasons habitat destruction is such a huge deal for so many species. And then finally, behavioral isolation. All right. Lots and lots of species, this is primarily animals, of course. Plants don't behave in the same sense that animals do. But a lot of animals have very, very finicky behaviors that are required for reproduction. The songs, that birds sing to attract mates. The chirping that frogs, and a lot of other amphibians do to, call mates over them. And also a lot of reptiles, like geckos. You'll hear geckos on warm, wet nights, in the summer, calling to each other. That's behavioral stuff. And in the case of the birds, the frogs, the geckos, any number of animals that have complicated behaviors that are responsible for reproduction, they tend to come up with new versions of these, some mutation or sometimes a behavioral change. Just, like in birds, they learn songs and they change their songs based on local influences. And if those changes lead to differences in mate selection, and this has been shown again for a lot of species, we can end up with gene pools that are isolated from each other where, the birds with the triple chirp at the end of a deep note, that group of blue jays is not reproducing with blue jays that have a quadruple chirp, call without a deep note at the beginning because, the birds, the deep note birds are not triggered, they're not triggered to reproductively engage with another individual if they don't hear that deep sound. And vice versa for the ones with just the 4 high chirping sounds in their call. I just made that up with blue jays, but just as an example, because this is a big deal and why we have so many different species of birds. They have behaviorally isolated each other over vast amounts of time. Oftentimes that's compounded by temporal or habitat isolation, and then chromosomal changes have appeared sometime over time, like between chimpanzees and humans, this aneuploidal event, and that has cemented the isolation that was here because of these factors to start with. And so now we have western blue jays and eastern blue jays, which are completely separate species. All right. They didn't draw as much in the old days. If you were a student of biology, early in the last century, your textbooks would have been full of words and almost no images, so you had to use your imagination a lot more than we do these days, to turn words into pictures. But here's one of the few images that was in Darwin's Origin of the Species. And, it's a branching cladogram. And remember in lecture I said, these branching diagrams, this branching diagram sort of way of thinking of biology is really critical, that we start internalizing and thinking about what we know in these terms here regularly. Okay, I'll give you an example here and then come back to this. So here's an example that you should already know from Biology 1406. You know that we now broadly understand that life on Earth consists of 3 different domains. So here's 2 different hypotheses right here, the 3-domain hypothesis and the 2-domain hypothesis. Both of these have a lot of similarity in them. It's just kind of a terminological difference here. But the key idea is that we had an ancestral, group of organisms, way back here. These days we understand that these bacteria, 3.75 billion years ago, would have been classified as bacteria today and probably would have fit into this archaebacterial group right here, archaebacteria. All right, so that's the ancestor of life, and they're still around today. We've got, a surprising diversity of archaebacterial types all over the place. Long ago, there was a major branching right here where one, group of these archaebacteria went down a very different sort of evolutionary direction and are, all of the modern bacteria that we see today, including cyanobacteria, E. coli bacteria, most of the bacteria that we're normally familiar with, and certainly almost all of the medically important bacteria are over in this group right here. And of course, eukaryotes, what we understand is that eukaryotes are a highly derived, highly modified version of an ancestral archaebacterial, organism that lived somewhere back here in excess of 1 billion years ago and probably closer to 2 billion years ago. Sometime that's about where that branch point is back in time. This is tree thinking, okay? Going back in this same article right here, this is the Wikipedia entry on domains. Here's the same idea Let's see if I can make this come up a little bit bigger here. This picture. Oh, here's another tree right here. It's not what I wanted to show you. I wanted to see if we could make this picture just get bigger, but it won't do it. It's linking to individual things on it right here. Anyway, there we go. Oh well. Anyway, here's another tree that gives you a lot more detail on the bacterial, archaebacteria, and eukaryotic idea. So anyway, tree thinking. You already have some of this information. Keep thinking along these lines. So here we've got the case with the elephants. All right. And in lecture, I gave you a quiz question that had 3 different, elephant species, and that is the African savanna elephant, the African forest elephant, so this actually branches into 2 right here, and then the Indian elephant over here, which we can branch into other branches that are separate gene pools, the ones in Indonesia, the ones in Southeast Asia, the ones in India, are distinctly different pools. There's no gene flow amongst those populations anymore, but they're all reproductively still compatible with each other, whereas the 2 African ones have been proven to be reproductively isolated. Anyway, we've got a tree right here. This isn't complete. It hasn't got everything on it. It's just got some key, things on it. It doesn't even have the details I just told you about right here. Well, we could make a tree that was that detailed, but this one tells the story we want to tell, of an ancestor, that led to a diversifying group of species of elephant-like animals. These are all extinct, and these are the ones that are still around today, and here's the lineage, right? And that's speciation. And so what happened, how did we get to all of these guys right here? Well, some combination of all of this. Under these rules. Not those rules. These, for these rules right here. Sorry. Yeah. Oh, goddamn, where's the picture I'm looking for? These, based on these rules right here. All right. Let's see. What else have we got? Okay, so here's an example of, allopatric speciation, northern versus Mexican spotted owls right here. Here's, some more detail of what we mean when we talk about allopatric speciation. So I've talked about these ideas right here. Mutations, in the time lead to reproductive isolation. So, all of this stuff goes together. So we can have an allopatric event. One of these things separates a population into 2 separate populations. Then these things accumulate over time. Behaviors, different behaviors in different environments that lead to, that maintain reproductive isolation. And inevitably, if we wait long enough, the accumulation of chromosomal differences between the 2 populations, which then locks in the difference in species. So when 2 species have been different long enough from each other and have accumulated these sorts of changes, they can never rehybridize again. In this particular case right here, I don't know the details on these. It may be possible to get them to hybridize if you can get them to agree to mate with each other. If they do all of those things, show up in the same place, recognize each other's mating behavior, and do all of these things, they may make a valid hybrid, which is a bird that could be something in between both of these. But there's no good habitat for them between these 2 ranges right here. So the hybrids are never going to have their kind of own place. And so the idea that these will join back together to make one species may be possible. I don't know the detail with these. I could look it up. Y'all could look it up. But it's unlikely because of the geographic differences and the huge differences that have accumulated over time with these 2 different populations. All right. Here's another example of adaptive radiation. These are the honey creepers from Hawaii, and all of these birds are descended from its founder species, which is I think this is a South Asian bird. I can't remember. We'll have to look at where the honey creepers come from, but they're distributed well in Africa, which is pretty far away on the other side of the planet, from Hawaii. And so I think it's an Asian, they come via Asia, getting over to Hawaii. And Hawaii is one of the most isolated archipelagos on the planet, much more so than the Galapagos Islands, and also a little bit older. But this original species has adaptively radiated out to so many different islands, on the different islands, and to different habitats on the different islands, and these are all nectar drinkers. This is an insect eater. This is an insect eater. This is a fruit insect and, seed cracker right here, and these are nectar drinkers right here based on the long bills. Anyway, that is a case of adaptive radiation. So whenever it's possible, whenever especially you get brand new habitat that nothing lives in, whatever gets there first oftentimes adaptively radiates to create a whole bunch of species. Opportunity creates when opportunities show up, speciation tends to occur, is what we're understanding. All right, I've talked about aneuploidy, autopolyploidy, and allopolyploidy already in sympatric speciation. So, I won't come back to this, but here's some examples right here, of illustrations here where we've changed the chromosome count in some cells. So here's what it should have been. Here's, a cell that's got one extra copy of a chromosome in it. Here's one that's got one short. Losing a chromosome is almost always a deadly event. Nothing's ever going to happen with whatever this gamete goes towards. But this cell with the extra copy in it may still be able to function, in which case we've got the beginnings of a new species right here. And here's some examples just, walking through these steps. But I've already discussed this, so I won't keep, paying. But, walk your way through these examples right here. And we've talked about pre-zygotic reproductive isolating events right here. So here's the examples: temporal isolation, habitat, and behavioral again. Then we can have post-zygotic, all right? So, what is it that keeps two different species from hybridizing if their gametes get together? For things that live in the water or things that have random fertilization on land like plants oftentimes do, wind-pollinated plants, it's critical that we have post-zygotic reproductive isolation because the eggs and sperm can get together of animals or plants in a lot of environments. Think of aquatic externally fertilizing organisms that live in water, right? Plant and animal. So post-zygotic barriers are what accumulate between isolated gene pools. Hybrid inviability. If an embryo is produced, it cannot survive development, okay? So that's a post-zygotic barrier. Hybrid sterility, that's the mule right here. We can make a hybrid, but the hybrid can't in itself reproduce because the chromosome count is different, okay? And there's one more version in here that's not listed on here for some reason but it's in your book. And that is gamete incompatibility. Lots and lots of aquatic organisms that are closely related. Think of sea urchins. There's hundreds of different species of sea urchins and dozens often that live in the same environment. They all release gametes, into the water, and a lot of them reproduce at the same time for the same cues, but they don't hybridize with each other because the sperm of one species can't fertilize the egg of another species because the handshake proteins are different from each other. If you'll recall from 1406, sperm and eggs join because they fuse, if you will, a messenger protein and a receptor protein together, and if they click and they latch, then the egg and sperm can join together. So if we have a change in the gene that codes for one of those handshake proteins, now that egg or that sperm can't handshake with its opposite member because it's a mutant, Anyway, so that's the way that goes. Here's some examples of temporal, habitat, gametic barriers. So, this is damselflies right here. These are the reproductive organs of some insects, and they can't copulate with each other because their reproductive structures look different from each other. Arthropods have exoskeletons, including on their reproductive organs, and so they really are a lock and key sort of thing. If the lock and key don't fit together just right, they can't reproduce, and so that's oftentimes a barrier to gametes getting together, structural things. Same thing with pollinators and flowers, right? All right, so we do have hybrid zones. They do exist, and it can happen. All right, and what happens when two closely related species get in contact with each other and have an area where they might mate with each other? All right, well, we might have reinforcement. The hybrids are less fit or not fit at all, in which case that's called reinforcement, and we're reinforcing the idea that there's two separate species and they're not going to remix with each other. That's typical. The more different things are, the more likely this is to occur. For things that are extremely, similar to each other, really just two different populations of the same thing that have accumulated no major genetic differences or anything like that, or gametic differences, then the potential for fusion, is there, and they might completely refuse together, or we might have this sort of stable situation where the two distinct gene pools sustain themselves, but there's an area where they hybridize. And we see this in a lot of things. So dogs still exist, wolves still exist, wolf-dog hybrids persist and are out there and maintain themselves, but they're not becoming the dominant thing. And so we have kind of a stable situation. Even more so between, say, dogs, coyotes and wolves, same sort of deal. Coyotes, dogs, and wolves are all the same species. So, they're not joining back together into one species. They're being maintained as distinct species because of behavior and size and smell and all kinds of things that have to do with reproduction and whether they view each other as potential mates versus just something to eat or a competitor. Final idea. This is, I think, this is our last idea right here, and this is the rate of speciation. Okay, the process of mutation and selection can theoretically occur continuously, and so the idea is that a species inevitably is going to change over time. It doesn't mean that it has to change externally. Cockroaches from 250 million years ago look remarkably like cockroaches today, but you can bet that the handshake proteins on their gametes back here 250 million years ago are not going to be the same as the handshake proteins on their gametes today. They will have evolved on that, if nothing else. The proteins that protect them from toxins in the environment as toxins have changed. So the point is, change does occur. All right. But remember that idea of adaptive radiation. When there is an opportunity, new land appears, a competing species goes extinct, the environment changes radically so that populations of everything are shrunk down terrifically, and something that can do really well in that new version of the environment, colder now, hotter now, drier, whatever, has a chance to really, take kind of command of that area. That's what we mean by adaptive radiation, and those adaptive radiations create a pattern that we see in the fossil record called punctuated equilibrium. Long periods where things are relatively calm, then something changed, oftentimes an environmental upheaval, volcanism, meteor smashing into the planet, ice age, whatever. And then we have a death of a whole lot of stuff, and then we have a whole bunch of new species, appearing, a new genus and new species from that genus, whatever, and again and again and again. As we go back in time, we see this happening where whole new groups, radically new groups of things are showing up. As we get closer and closer to the modern times, it's typically more at the species level where we see these diversifications, rather than a whole new group of animals with an entirely new body plan. But we continue to see in the fossil record these hiccups, of relatively quiet followed by major change, and those are usually associated with major perturbations in the environment, whatever the source of that perturbation. The fossil record is called punctuated equilibria, and each one of these novelties is an adaptive radiation event. Speciation can be, slow and gradual, as we're showing right here. Or it can be, relatively, dramatic. A single mutation radically altered the bills right here, but there was food that they could take advantage of with this altered bill, and so that actually worked, and it was desirable, and this population, rapidly diversified. Selection kept favoring a longer and longer bill right here, while these guys went in a different direction. There's no one answer. It totally depends on the species in question, the random mutations that occurred genetically along the way right here, and the power of the selective environment. If selection is extremely fierce, we tend to get this kind of punctuated equilibrium where we radically sculpt, a new population rapidly. If selection is weak, we have very, very gradual speciation. It varies based on conditions. There's no one answer. And that is a conclusion to this 1 hour long summary of the material in Chapter 18.